Hi everyone, we'll just let everyone come on. We've got people quickly coming on now. We're up to sort of 25, but in the first 1 second, so it's rapidly going up. I'll just let everyone, I'll give everyone about 1 minute or 30 seconds just to join us. But welcome to our 8th session for our biocontrol workshops. And I'll give people 10 more seconds. If you want to take the time, what we'd really encourage is if you could introduce yourself in the chat box there. Please say who you are, where you're from, and share something interesting if you've got the inclination. And I've also just put the agenda up there for you as well, so you can see what we have in store for you today. Just remember, the main way we're going to interact today is through the Q&A box. So if you've got questions, please put them in that Q&A box, and we will ask the speakers from there. Please use the chat if you want to share any information. So I'm going to start the session. I'd like to welcome you to the session 8. Time flies because this has been a 6-month series and this is one of our final workshops of the session. It is around the important role of biocontrol and integrated pest management. We've got a very busy agenda ahead of us. We have 3 speakers, 3 experts, and we're going to have examples drawn from the Philippines, from Brazil, and from Cambodia. So we're really lucky to see that. And there'll be plenty of time for questions and answers as well. Just remember, the main way we do participate in this session and the way we interact is through the Q&A box. So please try and ask your questions in that box there. That's really important because it helps us to run an efficient session. But we really want to hear from you as well through the chat. So if you want to share your work, your research, thank a speaker, please put that in the chat and make sure you introduce yourself to everyone as well. Now, just a reminder that the ASEAN Action Plan and Fall Armyworm Biocontrol Technical Workshop Series is a 6-month series. We're almost at the end. It's been supported by Grow Asia, CABI, IPM Innovation Lab, and many other organisations and institutes and the private sector from around the world. We are very delighted and feel very privileged to have all those experts who are talking today, but have also other experts who have spoken in previous workshops. So it's a little bit sad to get to the end, but it's also been quite an achievement. If you would like to give us feedback and questions, we would really like to hear what you're working on biocontrol and even what you thought of the biocontrol workshop series. And you can do that by going to ASEANFAWaction.org. Also, if you want a certificate of participation, you must subscribe to that forum and either ask a question, share something interesting about biocontrol, or note something you found useful in this series. And just to tell you how you can do that, you go to the website, you then click on community, click on forum, and then you can comment in one of the forums called Biocontrol for Fall Armyworm. And we already have quite a lot of interaction happening there. Now, as I said, this is the 8th session. We're almost at the end. We will have one more integrative workshop where we pull together a lot of the strands, and that will be held around August. We are still to finalise the details for that, but as soon as we do, we will email those to all the registrants that have participated in this series. Now, I'm going to launch a poll. It's not a scientifically rigorous poll. It's just to get us warmed up, and I've got some kind of different questions there and some more serious questions. The first question is, what is your favorite insect from the following list? And I'm kind of keen to see what you choose. 2 is, what stakeholder group best describes the organization you work for? And 3 is how many ASEAN fall armyworm action biocontrol workshops have you participated in? So I'm keen to see who's joining us today. So I can see just for your information, your favorite insect. Oh, it's a bit of a competition here between the ladybird and the fall armyworm. So if you are liking any of those other insects, get in there fast because they are behind by quite a way. You can see it's a hot competition there. Stakeholder group best describes the organization you work for. We have got quite a lot from research university, private sector and international organizations. And I will give you the results soon of how many ASEAN fall armyworm action workshops people participated in. I'm going to give people another 30 seconds to participate. And I'm kind of keen to see which is the favorite insect here. And Pranab, do you want to launch the end the polling and launch the results? Okay, I hope everyone can see those results. Ladybird was a bit of a clear winner there. Stink bug wasn't very popular, so that's a little bit unfortunate for them. And I'm surprised there's a lot of people that really have a favorite fall armyworm preference. It's nice to see. Embrace our enemy. Number 2, what stakeholder group best describes the organisation you work for? Actually, that's really kind of interesting. We've got a lot of government people. Thank you for joining us. Research university organisations, good private sector, but also quite high level of international organisations. And welcome to the students in the room as well. And this is quite exciting because 39% of you have actually participated in 5+ of these workshops. So, and another 35% have 3 or 4. So that's amazing. A lot of you have followed us through the whole series. So thank you for participating in this. And yeah, I hope you enjoy our very last one today, which I'm sure you will. So I'm going to stop that poll. And I'm going to move to the next screen. And this is my chance to introduce our first speaker, Mario Navasero. He is a scientist with the National Crop Protection Center at the University of Philippines, Los Baños. He has many, many years of experience in IPM, and he has a very active research team around him that I am in contact with as well, and who are also working on fall armyworms. So I'm really excited to introduce you to Mario, and welcome. Mario, are you on the line and can you hear us?
Yes, Alison. Good
afternoon. Before we start, what was your favorite insect out of that list? Did you have one there?
Silence. I'm working on silence.
Oh, okay. Right, great. Okay, so I'm going to let you, I'm going to stop my share. Could you load your presentation up?
Okay. Okay, good afternoon. Can I start now?
You can, please. You're welcome.
Well, I'll be sharing my some of our experiences in the biological control research here in the Philippines and try to relate it with our present problem on POW. So what will be some of these experiences that we can actually use in terms of POW management. So Ms. Allison gave us a guide questions to answer for our presentation. So I'll try to answer them one by one through the following slides. So what's the role of Biocontrol? Let me first start with the current problem that we have now in the Philippines. I think fall armyworm, with the latest data given by the Bureau of Plant Industry, we have around 25% infestation of our corn growing areas. And the problem now is that the previous insecticides being used for fall armyworm especially those used for wall application and basal application. We have fipronil and carbofuran. We have found them to be not working anymore. Even from the start, when fall armyworm arrived, farmers are already complaining that these chemicals are not effective anymore. And uh, initial talk with farmers for sweet corn, because sweet corn is considered a high value crops. The regular spraying is every 3 days, but when fall armyworm came, spraying increases 3 times a day, from three days interval to 3 times a day interval. So they usually practice cocktailing using different insecticides, but if you will look at the initial list, most of them are actually belonging to the old groups of insecticides, which we have documented in the case of onion armyworm to be not working anymore, like pyrethroids, carbamates, and organic phosphates. To start with, actually, usually the first line of defense when we have this outbreak is to find out the more effective insecticides, and actually have done that. We were able to find out which of these chemicals are effective, because the government, our fertilizer and pesticide authority, came up with the what we call this list of insecticides with special use permit, and we found out not all of those given special use permit were effective. So we have here identified the effective ones. However, we have serious concerns regarding this. Although we can say we can effectively control fall armyworm with these insecticides, but it has been our experience. Even for example, with our probably with diamond back moth and eggplant protein salt water, in 2007, 2008, they came up with the very effective insecticides belonging to the diamide group. Into the 3 years, we have already documented full-blown resistance. So in this case, in some areas where the government provided insecticides, and in usually government agencies provide only 1 or 2 brands of insecticides, and in some areas that we have observed or went to, some farmers have already observed declining efficacy of this insecticide. So this is very alarming, because among those insecticides that we found effective, 4 active ingredients belong to one group, to one mode of action, and all of these are actually, when we know for a fact that resistance, there will be cross resistance within the different active ingredient belonging to the same mode of action. And another concern is that we have already documented, or even the seed companies are telling us that some of their single cry protein events of GM corn are already susceptible to foul. So some companies are already withdrawing this single event product. So let me just start sharing with you our experiences in handling some of this outbreak using biocontrol agents. For example, this outbreak of cutworm in Pangasinan in 2003, if you will notice, we have around 4 fully grown cutworm in one plant, eggplant. And fortunately, during that outbreak, farmers are already complaining they cannot control the cutworm with the insecticides they were using. But fortunately, when we went in the area, we already noticed that a few individuals of cutworm who already been infected with NPV. So what we did is ask these farmers to collect those infected with NPVs and spray it again on their field. So 1 week after those, after spraying those NPVs, they already saw cutworm on top of leaves, all are infected. So from this simple field demo, we try to exploit what is existing in the area. So in this simple field demo, the farmers were easily convinced with regards to the effectiveness of NPV for cutworm. So we asked them to collect dead cadavers of cutworm, store them in mineral water plastic containers, and they can put it in the ref or buried it in the irrigation canals, at least to provide stable temperature. So, and from these collections, it is the same preparation that they use to control or to spray other fields infected with carbon in the area. So, because of that experience, the farmers asked us if we can train them to produce their own NPV. So we came up with a village level training on mass production of NPVs, and this actually became a popular one in the area. As a matter of fact, it was picked up by one of the company, Northern Food Corporation, who is engaged into processing tomato. So they invited us to also teach their technicians and farmers to how to produce these NPVs. Not only cutworm NPV, but also the Helicobacter NPV. So we tried to also put demos in their areas, comparing with their crop protection practices, using purely NPV and those in their practice using different insecticides. So they were convinced regarding also the effectiveness of NPV. So another outbreak that we were able to encounter during here in the Philippines, it was in 2005 to 2007, the outbreak of the coconut leaf beetle brontispa. In other countries, it took about 4 or 5 years before the outbreak was put in control. But here in the Philippines, it took only about 2 years, because during the early stage of the outbreak, we were already documenting several natural enemies. And as a matter of fact, in about less than a year from the report, we were already observing some trees recovering from the attack of brontispa. In this particular case, if you will notice, we have here Chelisoches morio. This is the very first natural enemy that we concentrated on. We tried to develop a mass rearing technique using indigenous materials, coconut fir dust, reusable plastic containers of soft drinks, fabricated cages using plastic, and we use what we call this fingerlings primers, because in mass rearing of earwig, they are using dog food, which is very expensive, and you still have to ground it. While the fingerlings, fish fingerlings primers, they're already powderized. So we also introduced village level mass production, and some farmers use this in one area in their field here, and they were also convinced with regards to the use of earwig for the control of brontispa. And this is also used for the control of pest of bananas. The most recent probably is, you have heard the news of outbreak of coconut scale insect here in the Philippines. Actually, there were 3 major areas that outbreak happened. In Luzon, it took about 4 or 5 years before the infestation subside, and in one island province, Basilan, it took about 6 to 7 years. But in Zamboanga Peninsula, where we immediately introduced the parasitoid that we encountered in Luzon, it took only 2 years. After 2 years, the problem was already under control. In Zamboanga, it took about 7 years because they came up with about 7 rounds or 4 rounds, 3 to 4 rounds of trunk injection. In Luzon, they attribute it to the typhoon that came in during that time. But actually, we've been saying that this parasite is already in control in Luzon during that time. So let me share with you a very specific experience. This is in corn. In 2000, BT corn was introduced in the Philippines, but we have also done BT corn being planted by our farmers. So what alternative can we provide to these farmers, even for those who are, because at that time many are still against the introduction of GM corn. So we thought of exploiting the use of NPVs, and also we have already we're already successful in mass producing Orius stantylus, predatory bug, which is very effective for corn borer. At that time, that was December when we are about to start our project, and the GM corn are already existing in the field, and we found out that in GM corn fields, the population of Orius are already high. Remember that one advantage of GM corn that they are claiming is that it actually encourages the buildup of natural enemies. And true enough, in that area, we were able to find out that the natural population of Orius, the predator, is already high. So the farmers actually challenged us because during that time, they say they cannot produce green corn December, January, February, because it is always being attacked by corn borer. So what happened really is we told them that they will not have any more problems with corn borer, because our hypothesis then is that the natural enemies from BT corn will only transfer to those non-BT corn once they start planting it. So, and that's exactly what happened in our experiment. So during this is our experimental plants, and when we took samples of the tassel, we found about 10 to 25 individuals of Orius. So this particular experiment, which is supposed to be tested for Helicobacter NPVs, for the effectiveness on lepidopteran pest. So without even any intervention, this particular cornfield was able to harvest even without using any control measure. So one way, what if we don't have Orius in the field? So there is another simple technique that we were able to develop. We use uray garden. Uray is actually what we call this? Let me. It's a weed. Amaranthus. It's Amaranthus spinosus. Remember that in natural condition, you won't be able to produce so much Orius in Amaranthus spinosus if this predator will depend only in terms of the natural occurrence of their prey. So we were able to develop another technique of fast-producing flower mite. With the flower mite, we put them in the fluorescence of Amaranthus. So this served as food for the Orius. So in that technique, we were able to produce around 100 Orius per plant with this technique. So another technique that we are recommending is to advance the planting of Amaranthus spinosus on the borders of the field, so they can enhance or encourage the initial establishment of Orius. So recently, probably another technique that we can use for the control of fall armyworm is the use of botanicals, and lately we have already done also some field trials, and we found out about three or four botanicals that are also working for fowl. So and this one is very matrin is very exceptional in terms of its effectiveness for fall armyworm. The neem oil is not that effective anymore for fowl. So the question now of how we can overcome the apprehension of farmers in using some of these biological control agents or biocontrol technologies. In the examples that we shared to you, we are actually emphasizing here the participatory approach that we did and the actual demonstration and to show that the biocontrol agent can really control their pest. Then we also try to develop the village level production technique for this different biocontrol agent. And of course, exploiting the existing natural enemies in the area. So with regards to possible researches that should be done, fortunately, here in the Philippines, the government is funding a lot of researches, especially those on biocontrol. So these are in various level of development. And in one of those researchers have already been producing metarhizium, and according to the researchers, they are already giving out this to farmers in one region. So we have here several researches. So what is the gap now that we are considering here in the Philippines in terms of the control of armyworm? We have several species of armyworms. We have the true armyworm, Ithena separata. We have another one. This is Ithena separata and the black armyworm. We have occasional outbreak of this, but the outbreak doesn't last long because we have very good diverse natural enemies here in the Philippines. So the challenge now is at this stage, even 1 year after the introduction of PAW, incursion of PAW here in the Philippines, we start already documenting several natural enemies. So the challenge to us is how to make these native natural enemies that are now having new associations with PAW, how can we enhance their performance, just like the natives in the native species of fall armyworm. So the challenge for us is how can we enhance these natural enemies so that ultimately, because to be able to manage PAW, ultimately we have to make these natural enemies work. And I think in many cases of outbreak here in the Philippines, even introduced species, in 3 to 4 years, there is a natural decline in population of this outbreak species. So we are also expecting this in fall armyworm as long as we will be limiting control practices that may encourage the development of these existing natural enemies now here in the Philippines. So that's all, I think. Thank you.
Excellent. Thank you so much, Mario. Brilliant examples, and it's really good to see. I think some of those lessons that you learned, really interesting how you worked with farmers and to actually also have farmers really interested in part of the process. I think that's got some good lessons for us all. We have lots of questions for you, so I hope you're ready.
Okay,
Probably won't be able to answer all of them, but here's a question for around, I think it was back to the start of your presentation around the resistance and the use of pesticides. Are fipronil and carbofuran not effective because the fall armyworm has grown resistance to it, or are these just not the right ones to use to fight fall armyworm?
Well, remember that the fall armyworm is an introduced species here in Philippines, and even in many parts of the world. And I think some of these fall armyworm that came from the Americas may be carrying with them already this resistance to some of these insecticides, because when you review the literature, there are already many reports of resistance even in the Americas for several insecticides. So we suspect that maybe the population introduced into this new areas have already their resistance already. coming from the origin.
That's very interesting. And we had a bit of discussion on that 2 weeks ago in our resistance management workshop. And you mentioned it today around these sort of single trait BT corn.
Yeah.
Do you think, is it time to sort of make sure that they're withdrawn maybe from use to sort of avoid resistance? Because there is cases of that in Brazil, isn't there? Resistance building, or has been found against in these single trait BT maize varieties.
Come again?
No, I guess what I'm asking is, you talked about the single trait BT corn and the potential resistance for
Yes.
armyworm, and you said that there's still some of that in use in the Philippines. Do you think it's time, or do you think there's some pressure to withdraw that then from use?
Actually, some of our staff that went in those areas received information even from the technical people from the companies that they are actually withdrawing some of these products.
Okay.
And they are now going into pyramided
Okay.
GMO.
Excellent. Now, here's a question here. Nowadays, farmers give attention and interest for biological control methods, but how can we make them take up biological control methods with no fear? Encourage them more. I think you gave some tips today about including them in the process, but how do you really encourage farmers more? Are they really interested in trying out biological control solutions?
Well, if they are convinced of the effectiveness of the biocontrol products, as a matter of fact, one of the projects that I have listed in the list of projects that I presented actually are claiming that farmers are clamoring for some of the biological control agents already. So the demand, I mean the what we call this, they're already aware, and especially when comparing this to the use of insecticides, which of course is very expensive, and the government is planning to sell these biocontrol agents at a very low price. So I think it will not be very hard to really convince some farmers to use this biocontrol.
Okay, that's good to hear. Here's a question here. There was quite a bit of interest in this. What species of Oreos are you referring to?
We have here Oreos tantilus.
Okay, there we go. There's an answer for that. Now, here's quite a specific question. I'm not sure if you'll be able to answer this, but what dosage of potassium salt of fatty acids was found effective?
Okay, I was not able to emphasize that. Potassium salt fatty acid is effective in terms of when we talk about damage and number of larvae. But I forgot to emphasize in that slide that potassium salt of fatty acids is phytotoxic. When you look into the plant height, it's the only treatment in our experiment that are what we call very short, because the internodes tend to be what we call this the internodes are very short, so the plants appear stocky, and even the yield when it comes to yield, they produce small ears.
Mm-hmm. Okay. Here's sort of a more technical question, but I'm sure you can help out. Is it good to use the Davis scale, which is actually for plant damage, for understanding for pesticide efficacy, to categorize products into efficacious and non-efficacious?
Yes, because I think Davis scale is now the standard method being used, not only here, I think all over the world now. And we can actually relate this also on terms of count of larvae. And although we haven't done that, but in terms of our graph or data, we both present Davis rating and also count of larvae. And there is also what we call the same results when we look into the what are the effective and what are not effective insecticides.
Okay, great. Here's a question. Did anyone try releasing Trichogramma effervescens to parasite the eggs of fall armyworm?
I think during the initial incursion of fall armyworm, they tried it in one region of the Philippines, where we first encountered fall armyworm in region 2. But it seems uh, it didn't work for fall
Okay.
armyworm.
Oh, that's interesting. Here's a technical question here. Well, I think it's a useful question. What is a whirl application or a wall application?
Wall application is actually your usually this is used for products that are formulated as granules or powder. You put them right there in the wall of the corn, the
Excellent.
drawing
Thank you.
point.
Thank you very much. No, it's good we have a range of people here today. So you can see we've got sort of more basic sort of technical questions through to very detailed technical questions, and you'll see those soon on the Q&A board. Thank you so much, Mario. That was a very good presentation. I love the examples, and there's lots of lessons there to take on board. I'm also really excited to see how much research is being done and what you're doing now. And we're looking forward to seeing some of the results, I think, of your laboratory and research team. If you could take the time just to jump on the question and answer board, there's actually 19 open questions there for you that we won't have time to ask now. If you could answer some of those, type the answer, that would be most appreciated.
Okay.
Thank you so much.
We'll try.
Thank you
Thank
so
you,
much,
thank you.
Mario. Thank you.
Thank you.
And I'd now like to introduce our second speaker today, Dr. Agenor Mafra Neto, and he is a chemical ecology researcher and entrepreneur in the entomological field of insect chemical ecology. He's the CEO of ISCA Technologies, a company specializing in the development of semiochemical solutions for pest management, robotic smart traps, and nanosensors, which sounds pretty exciting. So I'm excited to see your presentation, which I know is going to sort of take us in different directions and looking at big scale and small scale farming, maize farming and fall armyworm. So welcome to our workshop, and please start.
Thank you, Alison. Really a pleasure to be here today. Yeah, what is the role of biological control in IPM? And I'm going to talk a little bit about our experience in Brazil and a little bit also in India. So we have been working with fall armyworm for quite some time, and there are 6 major components to an IPM program that needs to be done everywhere we go. First is that to have a good identification of the pest. The other one is to be able to monitor and assess pest numbers and damage, but also the natural enemies that you have in the field. The third one is to have guidelines of when the management action is needed. So you can use preventative pests, you can prevent pest problems like avoid economic damage. And you should be using a combination of tools with different modes of actions like biopesticides, biological control, cultural, physical, mechanical, and chemical management tools. And once you take the action, you assess the effect of pest management. So it's really important in a program, an IPM program, any IPM program, to monitor either by scouting or using pheromone traps or any other form of monitoring. And pheromones and semiochemicals can help in the pheromone trap side of things. So biocontrol and biopesticides. Biocontrol is the method of controlling pests using other organisms. It relies in predation, parasitism, and other natural mechanisms. Biopesticides are pesticides derived from natural materials such as animals, plant, bacteria, and other minerals. And so there are 3 categories of biochemical pesticides. Biochemical pesticides, for example, pheromones and canola oil, baking soda, etc., microbial biopesticides, and plant incorporated protectants like GM crops that contain BT genes and express BT genes. So conventional control. Conventional control works by the grower taking the insecticides and chasing the pest with that insecticide. So it's really important that you have extremely good coverage on the in the field, that you cover every single part of the plant that you are trying to protect. The biggest problem with that is that it kills everything. It kills all the natural enemies, kills sometimes the pest. But one of the biggest problems is that we have been losing a lot of modes of action because insecticides drive the evolution of resistance. And the evolution of the resistance is very simple. You have some individuals in the population that are resistant. If you spray the same mode of action over and over and over, you then end up selecting for the resistant pest in the field, and you end up with a pest that is resilient to the application of that type of insecticide. The IRAC has 37 modes of action, including insecticides and different types of conventional insecticides, viruses, bacteria, etc. But what is really bothering me is that the Asian countries seem to, according to a report that I saw by this group, has only 7 chemicals belonging to 5 modes of action. So this seems to be a very, very small number of modes of action. And so it's really, really important that we have either a very, very strong integrated resistance management, or we bring more modes of action, or both. So we usually work with a different concept. Instead of taking the insecticide to the pest, we bring the pest to the control method. How do we do this? We usually focus on the mobile part of the population of the species, and usually we address the adults. The growers, usually they are worried about the larvae. So, but the idea is that if you kill one adult, for example, of fall armyworm, you're killing about 500 to 2,000 larvae, future larvae in the field. So how we do this pest control method, what we modify the behavior of the bug to come to the point source. We use sex pheromones for mating disruption, where males cannot find females, and we use attract and kill, where we use an attractant, the insect comes and manipulates the plant, the point source, and dies. I'm going to tell you a little bit about our experience in Brazil. Brazil, we have many different clients, but I want to talk specifically about the very large stakeholders in Brazil. We are working with groups that have between 400 to 600,000 hectares of crop production a year. And these people are producing, this is basically how they harvest and how they plant the new crop. They are harvesting the old crop, planting the new crop. So you have crop all over the year in this region here of Brazil. For fall armyworm, that's a huge problem, because fall armyworm has host all year round. So from January to December, there's hosts in the field. So you have populations that are always growing, and they become more and more difficult to control using conventional methods, and they are becoming resistant to many modes of action, including genetically modified plants. But the problem of Brazil is that you don't have only Spodoptera, you have several other different pests that are being used, that the grower is using insecticides in the field to control. So if you come with a solution, say mating disruption for Spodoptera, the grower still is going to use a lot of insecticide to control everything else in the field. So it's very important that you control a certain group, a certain cohort of pests. Otherwise, your product is not going to work for the grower in the field. They are going to continue to use conventional pesticides. So when we started working in Brazil in 2012, 2013, bringing new forms of protection for growers, for row crop growers. And during that interaction, I started working with the group of Bom Futuro. They grow more or less 600,000 hectares of different crops, basically soy, corn, and cotton. And after a couple of years, we started a center of excellence for semiochemical control for row crops. Together with Bom Futuro, both ISCA and Bom Futuro invest in this center, and that allowed us to bring products now that are being developed together with the farmer in a way that inserts the product inside the crop. These large growers, they want to go green. They want IPM with biopesticides, and biopesticides being biological or being semiochemical. So to control fall armyworm in Brazil, we found out that we cannot do only mating disruption. We have to do also attract and kill. We developed a formulation many years ago called Splat. We can apply by mechanically, and in Brazil we apply by airplane, tractor, drone, and you can also apply by hand. Splat is FAW, is a mating disruption for the fall armyworm. We apply this in the field, and this many dollops in the field compete with the females that are emitting pheromone, sex pheromone. Males cannot find the females, no mating. You end up, the population end up collapsing without a single drop of insecticide. That's the major advantage of mating disruption. The problem is that it's extremely specific, species specific. So mating disruption for fall armyworm works only for fall armyworm. And then we have another product called Noctovi. It's an attract and kill for noctuid moths, especially including fall armyworm. So this product, you put point sources in the field. Both males and females, especially gravid females that are very hungry, they come, manipulate the point sources in the field, and they die. And usually what we do is we sell the product. The grower uses 1%, adds 1% of an insecticide, and that affects control in the field, and also lasts for significantly longer than an application of insecticide. We did the calculations, and for about 1,000 hectares, a grower using only conventional crop protection would use a tank load, truck load of insecticide. With our product, they use about 200 liters, which is about 1% of what you have in that truck. So the integration of semiochemical solutions for the fall armyworm in Brazil is being done with Splat fall armyworm and Noctovi. And here you can see the application, the attractor application of the formulation. It's very sparse. You don't need to cover the entire plant. We did, this is a field trial where we did Noctovi by itself, and then Noctovi with Splat FAW for mating disruption, and the attract and kill for Noctovi and mating disruption at different doses. And we also use always the growers control method. You have to remember that these growers, they have access to every technology that exists for row crops in the globe. So it's they are working at the maximum. They extract as much as they can from these different techniques. So to go from, to show differences in yield in these crops is very difficult. Noctovi in Brazil, in these farms, is applied with motorcycles, and airplanes and tractors. And we use monitoring traps that water monitoring traps is a pheromone lure to monitor populations in the field. And here you can see that in that specific trial, in red, you have the control population dynamic. And then in the other colors, you have the Noctovi and Splat at V1, V2, V3, all the way to the end of the season. We also use the David scale to trigger applications of insecticides. So when you hit a percentage of damaged corn plants at the threshold of 20%, that triggers an application of insecticide. By using Noctovi and Splat FAW, we have been able to reduce the number of applications in comparison with the control that took 6. We were able to reduce to 3 applications only. But for the grower, reduction of insecticide sprays is great, is excellent, but for them, they need to look at yield and profit. So instead of us trying to figure out what is the yield, the grower actually harvested the yield, harvested the crop, and that allowed us to. Are we still? Am I still sharing or not?
Yes, you are.
Okay, good. And then check what is the how much of the product we have in the crop. What was the what does the yield in the crop, and as you can see, when you compare against the control, we got basically about 40% of larval reduction. We got 96% of trap shutdown, but this is the important part where the grower got about $92 extra profit at the end of the season. We did the same thing. Fall armyworm also attacks cotton and is extremely damaging to cotton because it goes into the flowers and the reproductive organs of the cotton plant. And when we do the same system to protect cotton, the grower ends up with a significantly high extra profit per hectare of about $1,000, which is more or less translates to more or less 5 extra bowls, 5 extra pods of fiber per linear meter of cotton. So all the things have been shown that semi-chemical control works in the field. We see increased populations of parasitoids, of other macroorganisms of control, natural enemies, and the system is working for these very large growers. What about small stakeholders? So we went to India together with our sister company, ATGC. And in India, the biggest concern that we had is that growers have very, very small plots. But the farms are a few acres large, so like 1 to 5 hectares. They have very high FAW infestation right now. And but the interesting thing is that in corn, FAW is the main pest. And that allowed us to go after products that can provide mating disruption. In India, we developed several different novel formulations for mating disruption, and we called them Cramit. There are 3 here. I'm going to show 3 forms of Cramit. One is a microencapsulated system with a type of organic glue that binds this microcapsules to the plant, so you end up with a very high longevity of these pheromone sources in the field, even though they can be sprayed with normal tractor and backpack sprayers. Here in these graphs, you can see in the bars, you can see the population damage or infestation that you have with a fall armyworm in the field. And when you look in this purple line here, you see the farmer's practice. So you can see that for most of the treatments that we have in this different in these different graphs, you can see that the grower has the highest populations and the highest damage in the field due to fall armyworm. The second one is the Cremit tablet, which is we infuse the pheromone into this nano porous matrices that are then put together, that are formed into pills. They drop that in the field, and the Cremit paste, which is very similar to the formulation that we have in Brazil. But in India, instead of selling this in very large containers, we have the grower using this in small toothpaste tubes. And this is the dollop that they have that they put in the field. Everything is done manually. And the Cremit SM, which is the microencapsulated system, we found out that we can do tank mixes with that. You can use the same tank that you're going to do an insecticide application, and the product works really well. Also works by itself, but it works really well with that. But I want to bring your attention to this. In Brazil, when we do our trials, we use a minimum of 30 hectares and as many as 300 hectares per plot. Here, we are using blocks of basically 5 acres, which is about 2 hectares. That's a very small plot, and I was very concerned that we wouldn't be able to get good control for this plot, in this plot. However, we went ahead, did the applications, and with the micro-encapsulated pheromone, we found out that it actually works. It works even better when you do tank mix with a few of the insecticides, soft insecticides that we had in India, and we can manage with 2 applications. You can manage FAW for the entire season, and there was a reduction of 40 to 50% of the conventional insecticide applications that the grower used to control fall armyworm. Those little tablets, we look at the number of tablets that we put in the field from 1,000 to 2,000 against the control or farmer's practice. Again, small little areas. And again, the tablets work significantly better than using the growers' control. And the application is very simple. You just drop the pill in the middle, on the soil or into the world of the plant, and then you're done with the application, and you repeat that in the middle of the season of V4 or V5.
1 minute left.
Sounds good. We have the Cremit P for FAW, where this one we already harvested, and we have between 11 to 25% of increase in control. So our conclusions are basically that biocontrol and biopesticides are extremely, they work together. They allow us to reduce the reliance on conventional pesticides. They bring more new modes of action for an REM program. They can control pests that are resistant to conventional pesticides, including GM crops, and they are an efficient way to control insect pests in small and large areas. They are easy to execute, and biopesticides and soft insecticides allow biological control agents to thrive. The things that I would like to see in a program like this is that the governments of this region, if there was a way to facilitate the registration of biopesticides and biocontrol agents. I would like to see if biopesticide and biocontrol agencies can be facilitated through the regulatory agencies. For example, EPA makes biopesticides much easier to register and takes less than a year, much less paperwork than conventional pesticides. I also would like to see the governments foster the registration of other insecticides with other modes of actions. We need to increase the modes of actions available for management of pests in the region, and promote safer, softer pesticides in the field. The other part, I think it was really well brought up by Mario, is to promote seasonship and get the citizens to demand for safer food and safer environment, promote area-wide IPM, the government promote area-wide IPM programs, and bring these pesticides, biopesticide and biocontrol agents to the growers by fostering entrepreneurial production of these biocontrol agents in co-ops, startups, the way that they are doing in Philippines. I think that's it. Thank you so much.
Thank you so much. And you have got questions. so hang in there. No, great presentation. I like the sort of reformulation of attracting the pest to the solution rather than the sort of pesticide to the pest. I really like that idea. You've got a few questions here, and starting with the first one, in attract and kill formulation, to kill insecticide is used. Is there a fear that insects can develop resistance to this insecticide over the years?
Yes, insects will always, if you don't control well if you don't have a good resistance management program, you are going to get resistant pests. So it's very, very important that you work with an insecticide that allows you to kill the insect. And there's a big advantage to our formulations, our attract and kill formulations, that we can, even though we are using 1% of the amount of insecticide that the grower would use per application, we are concentrating that in very, very small areas and into the formulation. So only the pest manipulates that. And in the case of Noctovi, the pest actually feeds on the formulation. So what happens is that these insects are getting a mega dose of the insecticide, dose that is much above than what would be needed to kill. And that delays tremendously the insecticide resistance. The other thing is that you can, the same way as you do with GM crops, where you can stack several different modes of action, you can do the same thing for this attract and kill systems. You can stack one or two different modes of action, and you can rotate them throughout the season, allowing you to avoid the development of resistance. But that's a very good question.
Yeah, good answer too. Here's a question. What actual active ingredients are in Splat and Noctivo?
So Splat FAW, the active ingredient is the sex pheromone of fall armyworm. And it's a product that we produce both chemically using synthetic chemistry in India, but also we are starting to produce that biologically, the pheromone, and reducing costs, allowing us to have very, very effective formulations at a very low cost. So biological, we produce that through fermentation or through plant breeding, genetically modified plant breeding that produce the pheromone itself. And then for the for Splat, we use for Noctovi, we use plant volatiles that are attractive to the moth, but are not attracted to natural enemies or pollinators like bees and ladybugs.
Okay, a couple. We'll just try and we've got quite a few questions, so we'll try and race through a few of them. Here's one. The paste method seems to be labor intensive and time consuming if you have to apply it one by one instead of just spraying it. Did you compare the cost of application for the products you mentioned versus the conventional method?
Yes, for that's one of the reasons why in Brazil, we do everything mechanically, by airplane, especially because of the cost of this product. So you have to remember, with these products, you don't need to cover the entire crop. So you can, with one pass of the airplane for Noctovi per hectare, you control the entire field. Whereas if you're using conventional pesticides, you would have to pass 4 times. So you're using much less material in the field, so it's easier. So even sometimes even manual application can be viable because the product lasts significantly longer than an insecticide application. In the case of Splat FAW, it lasts about 45 days in the field.
Okay. Here's an interesting question. I haven't seen this one before. Very interesting presentation. Thank you. Said the person. Could it be possible that the moth changes their sex pheromones as an adaptation to mating disruption?
It's possible, and that this process is called speciation. When a moth changes its sex pheromone, they have a completely different breeding system, and they become a different species. So it could happen, and all we need to do is to change the pheromone. But it's a process that in nature takes thousands of years. So we would have enough time to react.
True. We probably won't be around to see that, you and I, anyway. But are both
Definitely
products,
not me.
are both products, Splat and Noctovi, are they required to be used together, or can we just use one of them?
We can use one, and that was what we showed in India. We can use just mating disruption when the key population, the key pest, is the pest that you're targeting. If you have a mix of pests, then you need to use Noctovi, or you need to use a mix.
Okay. Okay, one last question, and then we're going to move on to our next speaker. But here's a question here. Many species of parasitoids eavesdrop on the sex pheromone of their respective pest and uses these volatiles to locate the pest. How can we be sure that the OCs will not attract the wasps, although it does not attract ladybugs and pollinators?
We have many years of this has been answered by academia, where they have shown that some parasitoids they track the pheromone, but are usually in the field we don't see that problem. We also, especially because there's a lot of space between these areas, between the applications, and so you end up seeing significantly more biological control in areas where you apply pheromones than in the areas that you use conventional control. It's possible that you might hit some of the species of parasitoids, but so far we haven't seen pollinators or other biocontrol agents dying, going for this or getting disrupted by this.
Okay, and one last question. What if the moths fly into the field and are already mated? Will the pheromone still have an effect?
The pheromone has no effect to mated females. And that's one of the biggest concerns that I had in India, where the areas were so small. So you have all these borders where the moth can enter. So if you, but nevertheless, it was really cool because we were able to see that the product actually worked there. So most of the females that are in the field, they are being originated in the field.
Okay.
Thank you so much.
Excellent. No, thank you very much. And I'm just multitasking at the moment and flicking through for our last presentation. But thank you very much for that presentation. Very interesting. And there's lots of questions waiting for you there to answer.
Wonderful.
I know, very interesting. And I'm sure when you answer them, those answers will pop up for the rest of the audience. So it's really important if you've got the time to have a look at that. And I really like the idea of something you mentioned at the start around thinking about how farmers are also managing other pests at the same time. So ensuring that you're not just thinking of one solution for them, but thinking about how they may manage that as part of an integrated pest management approach. So thank you so much. Really good presentation. Thank you.
Thank you, Alison.
Excellent. So we're now on to our last speaker today, Dr. Kim Ong To, and he completed his PhD degree from the Michigan State University in the US with a major in plant pathology and minor in sustainable farming and food systems. Kim is currently the Deputy Director of the Division of Research and Extension at the Royal University of Agriculture in Cambodia, and he also works on research and extension projects related to IPM at the university. And it's a pleasure to have you join us, Kim. And I just warn everyone this morning, he was frantic because he had no electricity but he got the presentation here and we're really looking forward to seeing it. So welcome, Kim.
Okay, Alison, could you allow me to share my screen because I have some
yes.
on the slide. Yeah.
Yep, I'm going to stop my. There we go.
Sure. Thank you. Okay. Good afternoon, everyone. This is Kim from Cambodia, and my presentation today is pretty much similar to the two speaker, but it's a little bit challenging to me for me to to get the presentation after the two brilliant research scientists. So so here is my content. So first question, I will just show briefly on the role of biological control in IPM, and then different biological control strategy in combination with IPM. But I also put some emphasis on fall armyworm. And then the last point will be on how to promote the adoptions of a biological control in IPM. Okay, so just quick general role of biological control in IPM. I think we can go back very briefly to the definition of APO. So we have different keywords here. So it's a combine of different techniques that are available for pest control. And then it's also need to be economically justified, and then minimize risk to human health and the environment. And then there are also some other points that for IPM that we could emphasize on the growth of crops, crop health, and then the keywords of agro ecosystem, and then also encourage other natural pests. So I think here it's biological control would have a lot of roles to play in here in addressing all of these key points. And then also it also could promote population diversity as well. If compared to application of chemical pesticide, I think it's a lot more destructive on diversity. And also address with the chemical residue, especially if we apply it before harvest. And then also reduce the risk of pesticide resistance. So it's the general viewpoint. And then I'm going to give some example that what we work here in Cambodia. So quick example on bok choy, and then another one on southern blight in solanaceous crop. And then I'm going to talk a lot more in fall armyworm. Okay, so here's a good example how we could incorporate biological control into IPM. So previously, some of my students and I conducted some research together in the insect pest control in bok choy. So we come up with different treatments. So I just want you to emphasize on the treatment number 4 that we use Bacillus thuringiensis and Bovaria bassiana in tank mixed together and applied every 5 days. And then another one, we have abamectin in early planting season, in early growing stage of the crops. But in the second half, we use Bacillus thuringiensis. And then treatment 6 also, we switch to region. Region is the fipronil. And then we also catch up with the application in the second half. So assuming that we have a bok choy here growing for 25 days. So at the early season, it would be good to use chemical control in our case. And then at the second half, it's better that we could switch to biological control because the plant is already big. And then if we continue apply more chemical control, and then we would have some chemical residue, because this is a very quick crop. And then usually farmer in Cambodia, they just apply every 3 to 4 days of chemical insecticide. And then some chemical insecticide, they have some restriction that we could not apply 7 days before harvest. And then it's how the biological control come to play in that, because they need to continue spraying because the population of the insect pest is very high. So it works out not excellent, but at least it helps reduce the application of chemical pesticide. And then another one, we work on a southern blight on solanaceous crop, and then we come up with different scenarios. And then you can see some type of biological control. For example, like chicken dung, trichoderma, chicken dung and trichoderma. And then all of this does not work very well to control disease. And then I think the best one is chemical control, azoxystrobin. But somehow it's also could help protect a little bit, delay disease a little bit. But at the end, all of this treatment plant die because we use very intensive inoculum in here. And then later on, we integrated grafting with biological control, and then it works. The efficacy improve. We switch the tomato and graft it on eggplants, and then apply biological control, and then you could see how the plants still could survive as compared to the plants without grafting. So in this case, what I want to mention here, if you want to promote biological control alone, sometimes for some disease or some insect pest, it's almost impossible. But if we have some way how to integrate it with other effective method, and then it would work well. Okay, and then for fall armyworm, I think in Cambodia we have very recent research, and fall armyworm it has been just appear in Cambodia 2 years ago in 2019 growing season. So we are very new on the experiment on this. But luckily, my team and I had a chance to visit Hyderabad, and we joined the fall armyworm conference before the disease outbreak. So we can collect a lot of information, consensus on type of chemicals, on application from different countries, ranging from India, Bangladesh, Myanmar, and Thailand. And we assume that those populations would be similar because they still continue spreading. So in Thailand, and then in late 2018, it's already occupied some of the province along Cambodian border. And then in 2019, it just appeared on some province along the Thai border in Cambodia. And then we conducted very quickly using information that we obtained from the conference, from the meeting over there. So chemicals control, imamectin and chloranthranidipole, I think they are top in the consensus at that time, and then we try to adapt and then modify a little bit to check the efficacy and then inform our farmer in Cambodia. So in 2019, we conduct an experiment in Battambang province, close to Thai border, on animal feed corn. And then later on in 2020, we conduct another experiment, but on sweet corn in the Mekong Delta. So these two locations is very different. Here is high, a little bit upland. Farmer grows mainly animal feed corn, but in the Mekong Delta, a lot of farmer grows sweet corn instead. So we conducted 2 different experiments. Okay, we also go through the standard experiment on fall armyworm, using pheromone traps, and then try to observe the population over time. And then, as you could see here, here is the picture in the for the experiment in 2020, that you can see the corn in buffer damage almost 100% and very heavily by fall armyworm. So very early in the season in 2020. And then for 2019 on animal feed corn, we come up with very simple experiment. So we have control, apply nothing, and then we have 2 application of imamectin. And then the treatment number 3, we have chlorantrienniprole alternate in 3 application. So chlorantrienniprole and then imamectin and then chlorantrienniprole before the plant reach VT stage to control. And then we have another option for biological control. I think it's a little bit not very makes sense for farmers who grow animal feed, because they do not find it's very useful to use a lot of biological control. But for our perspective, if that's work, at least it also provide them some options, and also maybe could reduce the application of chemical insecticide in the area. So the mating score, we use standard score 1 to 9. I think it's very standard, and it show in the manual of fall armyworm published already. And then you can see that chemical control work very well. And then for Bassiana, Bovaria bassiana, it just for animal feed corn, just not very different from chemical control, but it works well in the early season because we applied only 3 times. And then during the rainy season in Cambodia, a planting season, there are a lot of rains, and I think it's also play a very important role that those biological control would wash away in there. But the 2 treatment work well on animal feed corn. And here it's a percentage of plant also work the same thing, the same way that chlorantrienniprole alternate with imamectin would work best. Okay, and then here we have another experiment on 2020, and then fall armyworm reached the central part of Cambodia, and then we quickly set up the experiment. But here we increase another treatment. So we also consider the reduction because some farmers feel not comfortable with the application many times of a biological control, because it costs a lot of money. So we want to check if the reduce, how it would work. Okay. So as you could see here, chlorantrienniprole and imamectin alternate application work best, and then imamectin it's the second one. But Bovaria bassiana, I think it could help a little bit, not a little bit, but I think a lot in helping the reduction of the damage caused by fall armyworm on sweet corn. And then if you could check on this picture very quickly, as you could see here, control very damaged, I think almost 100% homogeneously. And then treatment number 1, 2 application of imamectin benzoate. Leaves look very clean in the early season. I think we also have another picture, but a little bit late, and then the plants were already tall, and then we could not see a very very distinct comparison here. And then treatment 2, chlorantrienniprole, imamectin, 3 application alternate, and then you could see here very clean leaves, some damage, but almost clean. And then compared to the buffer corn in between, they have very distinct damage on that. But here, when we check the application of Bovaria bassiana as compared to control, I think there could be a lot of improvement in here. Not very different in here, but if we compare to control, that's a lot different. And then for sweet corn, I think it makes more sense because it has very short like short planting date, and then only 75 days. And then there also demand for fresh consumption. And then could be some gaps or IPM or organic products. So biological control also could play a very important role in this case if a farmer want to grow the IPM or gap or or organic product for fresh consumer. It's not perfect as compared to chemical, but at least it provides significant control or protection as compared to the control. So here, yep, very clear that they have the ability to kill fallen mealworm in there. We also collect, like fruits, corn ear for yield parameter, length, weight per ear, and then score on the damage on fruits. And then it's biological control also could provide some protection as compared to the control, but when we compare to the systemic insecticide, it would be less. And we also check the break test to check if the different application might have effect on the flavor of the corn itself, but from our experiment we did not see any significant in terms of the flavors, the sweetness, So corn may be like this and like this. I would say taste would be similar, but they just affect very much on yield and then quality also as well. The marketable yield, I think it's the most important. And then to the last question, how to promote adoption of biological control in IPM, I think it requires a very systematic approach. I think first of all, we need to improve biological control value chain, especially in Cambodia. Not many biological control are currently available, and then it usually come on and off, depending on the value of the crops. And then they have other crops that use a lot of biological control, for example, like durian, some vegetables, and also black pepper. So area that grow a lot of those plants would have many options. But when the price decrease, and then they also are no longer available in the market. And then reduce cost. Biological control here is very expensive as compared to chemical control in terms of cost production. Promote investment, domestic production. I think most of our biological control were imported from India, some from Taiwan. I think mainly from India. And then we had very small demand, and then throughout the value chain it made the cost very high. And registration vary as well. So time for registration, cost for registration or re-registration, I think we need to improve that policy to improve the value chain and to reduce the cost, and accessibility as well. Improve people knowledge, awareness, both farmer and extension worker, private sector as well. Product branding and price incentive, I think very difficult for corn. And I think the only option that we could work on corn is on sweet corn. And then until they have a good branding and price incentive, so that farmer may consider using the biological control. Policy in research and education. It has to align well with the current policy of research and education. And then also improve of other cross-cutting issues like breeding program and other technological aspect that would be a cross-cutting issue, because as I mentioned that depending on biological control alone is not sufficient. You need to have, let's say, tolerant cultivar or other physical protection for the crops and many other. So it's the end of my presentation, and then this work was supported by we call USAID peer project, and then they collaborate with, we also collaborate with ERI. So they have the budget and we have the manpower. So we work together in conducting the experiment and the research for the 2019 and 2020. And this year, we do not have any activity anymore due to COVID, but some of my colleagues already have the Biocontrol, the parasitoid. They imported but parasitoid, and then they still raising them, but not yet.
Okay.
Yeah, not yet. Released to the field. Okay, thank you. I think it's a little bit long.
No, Kim, that was wonderful. Excellent. And you know what? It's always really valuable to hear what's actually happening across ASEAN countries. And we haven't had this amount of detail around what's happening in Cambodia. So it's great to hear this. And I'm just going to tell everyone we're going to be 5 minutes late today because I really want to ask you some questions. Presentation was really interesting. I've got a question here. Which strain of Bovaria did you use and coming from which insect?
I think I cannot recall specifically on the name, the strain. I have to check again, but it was imported from India, and it's a stain company. I think those who work in India might know about the name of the company, from Stain company.
Okay, great.
Yeah.
Here's another question, Dr. Kim. Do you have any data beyond week 4? How about combining the chemicals with biologicals and using the chemicals earlier in the season?
This is the question for Pak Choi, right?
Yep.
Yeah, I think we try to come up with different scenario in here, because in Cambodia, insects is almost resistant. I mean, for vegetable, leafy vegetable, the insects, for example, like diamond backmouth or flea beetle, they're almost resistant to or tolerant to almost every chemical. And then farmers spray a lot of chemical, and then they come up, it would be not very safe, because some chemical, it mentioned like 10 days or 7 days before harvest, they could not spray the chemical insecticide, right? If they spray, it would cause chemical residue issue. And then, so that's why we try the design that provide another option, that farmer could spray and provide some protection, but use biological control at the end, of the week, at the end of the week. Before harvesting. And then plants is already big, and then they also a little bit more tolerance to the damage. And like plants in the early stage, if we use biological control, based on from our experience here, it's very challenging to use biological control in early stage of the plantation.
Okay, I've got a quick question. What's always an interest to the what's always interested many people is to know what the infestation or how bad is fall armyworm this year. I mean, we get that question a lot. Have you seen the damage this year? Is it much different from last year? Because we hear varying reports. Sometimes it's better than last year and sometimes it's worse. What have you noticed in Cambodia or heard reports on?
Yeah, Cambodia, I think the farmer have just started planting. And then I've been in lockdown due to COVID in the city for a month already, close to a month already. My block, we are in red zone, and then people are not allowed to go out. Hopefully, if we can get out sooner, I could provide the answer, but not now. Sorry about that. Yeah.
No, that's
look. We will have another experiment on the using of we call conservation agriculture approach to control, and then let's see how they affect on fall armyworm in 2 different locations.
Okay.
Yeah, but it has been suspended, delay a little bit, and wait until the government open the city again.
Excellent. Yes, no, COVID has created problems for us all. But Kim, thank you very much. And I'm just going to, I've just rested on this slide here because I thought you virtually gave the summary of the webinar actually. when you gave your sort of presentation on this slide, you virtually summarized all the sorts of things that we need to work on and look at. So I just wanted to say thank you for that. And I'd like to say thank you for your presentation and bringing it together so quickly and under sort of trying circumstances. And I'm just going to close the webinar now because it's our last sort of workshop of this series before our very sort of integrative workshop in August. But thank you very much for your presentation, Kim. It was great to hear from Cambodia and what's happening there. And I'm looking forward to seeing the results of that conservation biological control research as well. So
Okay,
thank you, Kim.
Thank you for having me. Yeah, thank you for having me.
Excellent presentation. And I just want to finish very quickly. I'm going to make a very brief summary of today, a very rich discussion with many examples used to show the importance of biocontrol in an IPM approach to address fall armyworm and other plant pests and diseases. Mario pointed out the risks we already face with the potential overuse of pesticides before demonstrating the important factors in encouraging uptake of biocontrol solutions, such as including a participatory approach involving farmers, demonstrating the use and effectiveness and building capacity at village level. There was also a need to look at the power of what natural control solutions that already exist in nature and really harness that power, such as natural predators and enemies. I really like the reframing of the problem that Agenor talked about, moving from insecticides chasing the pest approach to a pest chasing the control method. A really nice way to reframe the problem into a solution. I really think it was great how Kim emphasised the need for multiple tactics to be used at the same time and the ability for biocontrol to allow that happen. And important to emphasise that strong role for government to encourage and support biocontrol from regulatory and social perspectives. And I think the last point that Kim underscored, the need to improve the biological control value chain, is extremely important. So a fantastic workshop and a really great way to end this series before our final integrative session later in the year. I'd really like to thank all the three speakers today. I'd like to thank all you people who have participated in this series as we've gone along. We've now had 8 of these, and many of you have actually followed the whole series as well, or at least 7 of the workshops, which is a great turnout. Please share your ideas and thoughts and get a community going on biocontrol. There's lots of interest there. Let's make it happen and talk further and keep the discussion going. I would just like to say thank you to Prana for helping out in this series with the technical sort of platform work. And I'd like to thank all the previous speakers as well. So thank you very much for joining us, and I look forward to keeping in contact. Keep safe, everyone, and good night or goodbye or good morning. If you could answer the poll at the very end before you leave, that would be most appreciated. Thank you very much.